NanoDrop technology combines fiber optics and surface tension to hold and measure small amounts of samples, such as nucleic acids and proteins. A small droplet of sample is pipetted directly onto the lower pedestal. An upper optical pedestal then engages the sample to form a column. The pedestal automatically moves to adjust for optimal path length. Shortening the path length eliminates the need to perform dilutions for most protein samples. Measurement occurs in seconds, and UV-Vis spectrum results are displayed.
Hi, I'm Lynn Keelhorn at the NanoDrop Laboratories of Thermo Fisher Scientific. Today, we will show a procedure for determining protein concentration using an innovative microvolume spectrophotometer, the NanoDrop 2000C.
The NanoDrop 2000C spectrophotometer uses an innovative sample retention system that relies on the surface tension of liquids to hold and measure microvolume samples between two optical pedestals without the use of cuvettes or capillaries. The microsample is placed directly on top of the detection surface, and a liquid column is created between the ends of the optical fibers by surface tension. This liquid column forms a vertical optical path. A xenon flash lamp provides the light source, and a spectrometer utilizing a linear CCD array is used to analyze the light that passes through the sample. Removing traditional containment devices such as cuvettes from the system has several advantages. Very small amounts of sample are needed for measurement, 1 to 2 microliters. Cleanup simply requires wiping the optical surfaces with a laboratory wipe, and the path length automatically changes in real time during the measurement.